everyone, morning, afternoon, evening. My name is Tracy Steinricken. I'm the president of the Australasian Mycological Society. Thank you for joining us today for our June virtual seminar. Before we begin, I'd like to acknowledge the traditional owners of the lands around Australia, New Zealand, and around the world on which we're all meeting today. I'm speaking to you from Brisbane in Queensland. It's the traditional homelands of the Turrbal and Yagara people, and I pay my respects to the elders past, present, and emerging. Our presenter today is Professor Treena Burgess. Treena's vision is to see an end to or effective mitigation of the impacts associated with biological invasions in natural ecosystems and forestry. Her research field is the biology, ecology, and genetics of beneficial and detrimental microorganisms in natural ecosystems, plantation forestry, and horticulture. There have been three major components to her research. Firstly, diseases of eucalypts with direct relevance to plantation forestry. Secondly, Phytophthora in natural ecosystems. And thirdly, complex biotic and abiotic interactions in tree declines. These are all collaborative programs involving research partners at national and international universities and government research organisations. In addition to highly cited academic outputs, her research in all areas has had tangible and demonstrable impacts on end users. Her current role as Research Director Institutes focuses on the implementation and operationalisation of Murdoch University research strategy to build on their strength in health, food, and the environment. Today, Treena will be talking about the Bridgehead Effect and how that relates to urban forests and invasive pests within Perth, Western Australia. She discusses the enormous impact of Phytophthora Cinnamon in the region and the increasing decline of trees within the city. If you'd like to ask a question or post a comment for Treena during her talk, you can do so by clicking on the Q&A button at the bottom of the screen. Treena will answer your questions at the end of her presentation. Then, at the conclusion of the Q&A session, at the end of the seminar, please just take a moment to answer the short survey to help us improve the delivery of this seminar series. So with no further ado, Treena, take it away.
Okay, I hope I'm going to operate this correctly. So hello everybody, and I'm very grateful for this opportunity for several reasons that I hadn't tell Tracy. I don't get as much time for research as I would like anymore, and this made me look at some data and actually get somewhere with something that I've been trying to do for a while. So I was quite grateful for that opportunity. So I'm going to do my acknowledgements of various people right up front, because often what happens is I get to the end and I say thank you and I forget to do it. So mainly I want to talk about the contribution of Diane White, who has worked for me for a very, very long time as my technician, and she basically has done all the technical work involved with the project. Peter Scott, Trudy Papp, Joey Hulbert, who was in South Africa, Thilo Massenbauer, who runs the DIDM's website in Western Australia, and Giles Hardy, who I've worked with for a very long time. It's also the staff at the Vegetation Health Survey at the Department of Biosecurity, Conservation and Attractions in Western Australia, various students that we've had in our group, and also a lot of other people, particularly around Australia, who have helped me out with this project. So I also would like to do an acknowledgement to country. So Murdoch University is situated on the lands of the Whadjuk Noongar people, and the university is actually on a very special site that was, because we've still got Banksia woodlands and swamp sites, and it was actually a place of knowledge. So it's very interesting that a university ended up being put there. So the welcome to country is really relevant and important to us, and I would just like to pay my respects to the Noongar elders past, present and emerging. So just because I'm a little bit of a fraud, I thought I would just remind everyone, I'm sure you already know, here are the fungi over here, and today I'm talking about Phytophthora, which is over here in the Mycetes, which is in a completely different part of the tree of life than fungi, but I guess they have mycelium, and we can talk about them in this context. So I'm actually going to do quite a lead in to where to the talk because I want to explain what has led me in this pathway to end up getting there. So it started when we started doing metabarcoding of Phytophthora out of natural ecosystems. So the project was actually aimed at exploring the limits of the distribution of Phytophthora cinnamomi. And we wanted to get samples from all over Australia, but we also realised that we wouldn't be able to do all the baiting and everything in order to detect Phytophthora in our laboratory. And so we decided to use a metabarcoding approach. And so that work, there was something like, I think, nearly 700 samples that we got from very unusual places around Australia. And we thought, well, this is really interesting. We got all the Phytophthora cinnamomi data, but then we also got all the data from the other species. And we thought, well, this is quite interesting and we should be able to do something with this, which we did. So the primers used for the amplification of the Phytophthora are Phytophthora-specific primers that were designed in 2012, and they're very, very specific to Phytophthora. They amplify every single species. There's been a lot of testing on them, and they will pick up a couple of phytopithiums, which are related, but pretty much nothing else. You have to use a nested approach just because the amount of Phytophthora DNA in the samples is usually pretty small. So you use it with the specific primers first, and then you increase it with using the internal amplification is with ITS6. We had samples that were from a whole bunch of ecoregions around Australia, quite 3 or 4 of them in Western Australia, quite a few in the up and down the eastern side of Australia, and then throughout Tasmania, fairly reasonably balanced between them. So what did we find? Well, we made some assumptions about whether a species was native or introduced. And these are only assumptions. We can't do very much about that. It is an assumption. But many of the agricultural pathogens are global and they've got really broad distribution. And then we've got a whole bunch of species that have either been described from Australia or only found in Australia. So it's all putative, but we considered that they were either native or introduced. And then we looked at things like habitat association, so whether it's in agriculture. So this is this sort of bars here: agriculture, agriculture and horticulture, agriculture, horticulture and nurseries, horticultural alone, native, N is native. So, and you could see that the introduced species were really strongly associated with samples that had come from an agricultural setting, and the native species were more likely to be associated from natural ecosystems. It's not very surprising. Across the different ecoregions that we looked at around Australia, it was pretty much balanced between native and what we thought reputatively native and introduced species. And the phylogenetic clades. So Phytophthora has 10, 11, 12, depending on who you talk to, recognised phylogenetic clades. And there are features that are associated with each of those clades. And for example, clade 7, where Phytophthora cinnamomi is, many of the species from that clade are associated with diseases in agriculture, usually in trees, like so woody plants, whereas clade 8 has a lot of species that are also associated with agriculture and tend to be diseases of annual plants. I mean, that's very broad. There is variation in that. So we got here and we looked at the data, and we actually found that there were very few patterns associated with environmental parameters, and we couldn't really link species to certain places. But I guess more for me, even though I like the whole concept of metabarcoding and I like the fact that you can look at a lot of samples and you can find interesting interactions, I still think that the technology has limitations. And one of the things that concerned me the most is that we detected species that have never been isolated in Australia. And we also detected 15 new species at that time. Some of them we've since found a few of them, but not that many. And so the big question to us was, are these species real or are they artifacts? And so this led me to go back to real data. And this is actually the project that I am doing at the moment. And I'm pretty close to the end of it. So I have looked at what is the current status of Phytophthora in Australia. And to do that, I've looked at every possible database and downloaded data from nearly everywhere that I possibly could. So they are all the Australian databases, but also the international databases that had any records from Australia. I've gone through every single publication that's ever had Phytophthora mentioned in Australia. And I've also downloaded any molecular data that I could from GenBank, and I've put together a really big database. I've got 8,000 records at the moment, and of those, nearly 3,000 are molecular records, so you can be they're pretty happy with the ID. You can see where these records are, and unsurprisingly, given that Phytophthora is a oomycete and needs water in order to be able to cause disease, it's found in general in the wetter parts of Australia and or in agricultural systems where water is being added into the system. So it ended up with 100 species that have been reported from Australia. So this was a little bit of a surprise. I didn't realise there were quite so many, because this is against a background where there's probably about 190 species described in total. And this is interesting because we detected 68 in metabarcoding, and we were a little bit concerned that we detected so many species, but actually when you can compare it to what is being reported in Australia, it's not really that different. Of those 80 species, 80 of them are known described species and 16 are new undescribed species. And as you can imagine, that's based on molecular data. So we have isolates that have been sequenced that belong to species that are yet to be described. And there's also what we call 4 designated taxa. That means somebody somewhere has given a taxa a tentative name, but it hasn't been officially described. But of those 100, I have decided that 10 are in doubt. There's no molecular data or molecular records associated with these, and their records are older. So, and I think that they were probably just names that were put on species because it was the best match at the time. And since then, there's been a lot more work and a lot more species described, and they may have been incorrect names. So, oh, I've got to move this. So, and just to have a look, show you where, you know, the climate, everyone knows the climate zones in Australia. Unsurprisingly, Phytophthora is found in the climate zones, which tend to be a little bit wetter and not in drier places in general. So this is a bit of data that has come out of that. So this is looking at the time and the cumulative number of species in Australia. So up and between the first species that was recorded in Australia was in 1900, and well, no, actually it was exactly in 1900, and that was Phytophthora infestans, which was first described in 1880 something, 2 or 3. And since then, we've had a whole bunch of species described, which is the wide circles. The closed circles are actually when the species was described. So there's a difference between when a species was described and when it was reported in Australia. So initially, all the species were described prior to being reported in a report in Australia. It's really interesting. More recently, many species are being described after the first isolates have been found in Australia. So it's actually a retrospective positioning. So, you know, if you think that they match over, it's retrospectively. So when a species has been described and we've done re-evaluation of existing isolates, we've found out that they belong to a new species. rather than the species that they had originally been designated to. All the early species that were recorded in Australia were introduced species, and they were found associated with agriculture. Everything recently is a bit of a mix between agriculture and natural ecosystems. And since 2009, 25 new species have been described in Australia, and of those, I think 22 of them have only been found in Australia. And I just wanted, at the very end, there's a bunch of species that are yet to be described. So there's species that we have isolates for and molecular data for, but haven't done the descriptions. And if you look at these species, where the dominant, the number of records that we're sort of getting and where they're going against the phylogenetic clades, you have a whole lot of very commonly reported species from clade 1, clade 7 and clade 8, which I've mentioned before are the clades that are often associated with agriculture, as is clade 2, and Phytophthora palmivora in clade 4 is another very common species in agriculture. So most of the reports, unexpectedly, because most of the diagnostics is done from agriculture, but we're starting over time to build up more and more data on things in natural ecosystems. And this is the same data, but looking whether the host that the isolate was recovered from was either an exotic host or a native host. So the exotic hosts, again, there's a lot of them clustering in clade 7 and 8, clade 1, a whole cluster of species in clade 2, which tend to always be associated with exotic hosts in agriculture. And then there's other clusters of species within the Phytophthora phylogeny that are generally found on native hosts. And then there's other species that have been found almost exclusively through water sampling. So, you know, this is the sort of data that I'm starting to be able to draw out of this very, very big database that I've put together. But this then takes me to, as I said, a very long introduction to where I wanted to get to talk about the Bridgehead Effect and where Phytophthora species are actually being found. So I kept on wondering to myself, okay, we're finding all these species. They seem to be everywhere, but are they everywhere? And how many of the introduced species are actually truly invasive, as in going into natural ecosystems, and how many of them are basically staying with their hosts in agriculture? And is this something that is going to change over time, or is there some sort of stability in what we're seeing and observing? So the concept of the Bridgehead Effect, it's about this concept that organisms, the invasion of organisms into natural ecosystems are actually, it's not direct, but there's some sort of stepping stone on the way where you've got some sort of build up of the species or of the organism within a particular environment. And there's been a lot of talk, particularly recently, about urban forests or urban ecosystems as being the environment in which invasive species have the opportunity to be introduced and adapt to a new environment and perhaps to new hosts before they move out of that environment. So there's quite a few papers that have been looking at it, because there's the other concept of, if it's true that the urban environment is where most invasions are originating, then we could use hosts within those urban environments as sentinels to potentially be target points for sampling, you know, the first sampling point to determine whether this really is the place where new species are arriving, and perhaps is a really good way of being able to detect them before they get out into natural ecosystems. And so, you know, we wrote this conceptual paper just a little while ago about urban trees and their as bridgeheads for forest pest invasions, and within that we included quite a lot of Phytophthora examples. And also did another little project. This was in Cape Town in South Africa, as using urban environments, particularly botanical gardens, as the place where new species are getting introduced and adapting to the environment before moving out into the natural ecosystems. And there's been a few papers around that concept. So what is it about an urban forest? They're a mixture of native and exotic plant species, so there's more potential hosts in an exotic environment than there is in a natural ecosystem. The environment is often not ideal for not, I've said both the plants use it, it means that, but what I mean by that is that the plants can be under stress in the urban environment, particularly trees, because of all the roadworks and everything that happens around them. So they're not necessarily in a perfect environment for them, makes them a little bit more vulnerable to disease. So it's a mixture, most cities, particularly in Australia, are a mixture of intensely managed spaces and conservation areas. So this gives a nice opportunity to have them next to each other, and you could have an exotic species building up its numbers within a managed space, and then it can either be planted into a conservation area because somebody's doing rehabilitation, so it's come from a nursery, or it could just move into the conservation area due to human movement. And then there's a lot of increased stress on those natural vegetation in the conservation areas because there's fragmentation, weed pressure, salinity, drought, etc. So they're not great environments often. And this isn't so much for Australia, but very much for elsewhere in the world. There's often very poor international and interstate biosecurity for plants for planting. And this has been a very big issue in the movement of Phytophthora around the globe. And so exotic species can arrive in the plant tray. They can establish in the urban environment, either in a garden or a park or on a botanical garden. They can adapt to new hosts, environmental conditions, and then they can be transported to rural areas and, you know, also probably in gardens, and then eventually into native forests. So this is my species, like how many species make it into intact native forests. So not into disturbed areas, not into remnants associated with cities, but actually into intact native forests. And because we've got such a large data set for Phytophthora in Western Australia, it provides an opportunity to actually really be able to look at this. And one of my inspirations for this was a paper that came out of Sweden, where they looked at the functional traits associated with the establishment of Phytophthora species in Swedish forests. And what they found was, when they did this very intensive sampling from nurseries into forests, and that's forests where there's human disturbance or they're completely natural, what they observed was that they had a decrease in the number of Phytophthora species that they isolated. So there were more species in nurseries, there were fewer species in the disturbed forests, and even fewer species in the natural forests. And this is separate from the ones that were found in the water. And they did a lot of sampling, and they found that there were various characteristics of these Phytophthora species which led them to be present in this sort of, so it's sort of an environmental filtering of sorts that is reducing the number of species that can successfully establish into an intact natural ecosystem. So Western Australia, we've had this huge impact from Phytophthora Cinnamon, which can turn beautiful natural forests with loads of banksia and various other understory species into quite sparse open forests with a lot of grasses and sedges in the understory. And here is Giles and Sarah Barrett at down in the Fitzgerald River National Park, one of the most beautiful places in the world, at a Phytophthora Cinnamon, where you've got the disease on one side, so it's actually a dieback front, where you've got disease on one side, and clean on the other side, and you can see the dead trees at the front. And after all those dead trees are banksias or various proteas, and they are completely removed out of the ecosystem afterwards, and you just ended up with lots of sedges and a few resistant plant species. So its impact has been enormous and continuing. And, you know, that's an example of one species that has definitely made its way all the way into natural ecosystems. And so how many other has had? So we actually tried to look at this in the urban environment. We had, again, very intense sampling where we had different types of parks within a jurisdiction within Perth, Western Australia. So it's in Joondalup. There was 236 soil and root samples were collected from 91 parks and natural reserves from a variety of hosts, mostly that they were all native hosts. So, but some of them were in managed parks where there was loads of water and lots of grass underneath of it, and a lot of them were just in conservation areas. So we detected 45 species across the sites, and we measured a whole lot of variables that we thought would be associated or linked with the Phytophthora community that we observed. But actually, nothing was linked. It was just pure admixture. There was no filtering between the grassy areas and the natural areas. And actually, even though, I mean, it was a really nice data set, it was actually a little bit disappointing to get these results. So here's an example. If we're looking at different park types, they're all aligned on top of each other, and they're basically all identical. If we looked at canopy cover, the Phytophthora communities were pretty identical. About one of the only things that showed any variability was whether or not two of the most invasive Phytophthora species, which are Phytophthora multivora and Phytophthora cinnamomi, were present. And if they were present, then you tended to have more other Phytophthora species. And if they were absent, you had a different suite of Phytophthora species. But that is pretty much the only thing that we observed. But when we thought about it afterwards and we thought about how we collected the data, we realized that we'd actually biased the data. So in a way, it was our fault, because even though we were collecting from different parks with different characteristics and different amounts of watering and stuff, we were always sampling from the roots of trees. So we'd put in an extra layer of filtering ourselves and by only sampling from trees. And perhaps if we'd done more random sampling, we would have seen different communities. So that was a little bit, well, it didn't go as well as it could. But so here is the data for Western Australia. So we're up to 64 species at the moment. Many of the IDs, so the Vegetation Health Service had thousands of isolates of non-Phytophthora species, and we have sequenced nearly all of them now. And so we've got retrospective IDs on them and their location data. And then we've got a lot of work from our own group, from agriculture and from the urban environment. So we've got like a really nice data set to start looking at where things are found. So in Western Australia, just to show you this concept of the Phytophthora clades. So this is a Phytophthora phylogeny. These are the clades, and this is sort of a little bit representative to the number of species that are in the clade. I've got the total number of species that have been described. I've put 207. There isn't actually 207 species described, but there's about 18 species that have got been designated as a taxa without a description. In Western Australia, we've got 64 species. This is all based on isolate data. This is real. We've got the sequence data for it. And of those, we think that 33 of the 64 are native to either Western Australia or Australia. And those designations are putative, but it helps us being able to interrogate the data. So what environments are they found in the, with these environments of different level of disturbance? So if we looked at nurseries and gardens, we have detected 12 species in that environment. If we look at urban bushland, there has been 36 species detected. If we look at orchards, so perennial agriculture, so horticulture and forestry, there's been 22 species. In annual crops, so this is things like potatoes and also pasture, you know, just in things that tend to be growing annually. There's been 12 species detected. Strawberries are in there as well, I think. In rural disturbed areas, we've got 38 species, and from natural ecosystems, there has been 31 species reported. But are these species introduced or native? So if we look through this again, so we're breaking this up into the nursery, garden, urban bushground, orchard, forestry, annual crop, rural disturbed and natural ecosystem, you can see that there's a very different mix between species that you would consider to be native and those that you would consider to be introduced in those different environments. So in the nurseries and gardens, most of the species are introduced. In orchards and forestry, most of the species are introduced. In annual crops, most of the species are introduced. In these urban bushlands and rural disturbed areas, it's a pretty even mix between native, potentially native and introduced species. And in natural ecosystems, nearly every single species that we've found is a native species, putatively native species. So I had to, you can't do a six-way Venn diagram, so I have added all the man-made environments, so all the agriculture and nursery into one highly disturbed environment, and then kept the urban remnants, rural disturbed and natural separate. And the first number is the number of species, and the number in the bracket is the number of native species. And you can see that there's, you know, quite a few species that are in man-made environments, only one of them being natural, and etc. So it goes around. And this thing in the middle with the 11 and 6 is the species that are found in all environments that may have probably made their way from being first introduced in a man-made environment, perhaps through the, you know, sort of as a bridgehead effect, through the urban or remnants, perhaps then moved into rural or disturbed areas and then potentially into natural ecosystems. It actually works a little bit better if I actually split this out, and then you can see that there are, that most of the introduced species are found in disturbed environments, with the exception of 5 that have been found in intact natural ecosystems, and most of the native species are found in the native environment, but some of them have also been detected in remnants as well. And one, funnily enough, has been just found in a grassy area in Perth, and it seems it's very closely related to another species, but that's a different story. So what we believe has happened is that we have had this introduction of a lot of species, but most of the species that you can consider introduced are pretty much staying in those disturbed agriculture environments, and that to date we have probably only had 5 species that have worked their way into natural ecosystems. So what are those 5 species? They are Phytophthora Cinnamon, of course, which we know about a hell of a lot about. I've spelt Nakoshiani wrong anyway. Phytophthora multivora, which was newly described in Western Australia, but we pretty soon realised that it wasn't native to Western Australia, so we described it in 2009, but it's actually now been found globally, particularly in the Southern Hemisphere, and we sort of think that it might have been native to South Africa and probably arrived in Australia a long time ago, maybe in the colonial times when they were stopping in Cape Town. Phatophtra Nakoshiani, which is a species which is distributed globally. Phytophthora hauseri, which is another newly described species, which is quite closely related to Phytophthora cinnamomi and is found a lot globally in tree crops, and it seems to be able to make its way into natural ecosystems. and Phytophthora Cinnamon, which is another species that we described, I don't know, maybe 6 or 7 years ago. It's very closely related to Cryptogaea, but if you look at the host range, like now they've been separated out, and if you look at the host range of Cryptogaea, it's almost exclusively agriculture. And if you look at the host range of Pseudocryptogaea, it seems to be trees, ornamental trees, and in the natural ecosystem. So there seems to be a little bit of a split of the host range. The characteristics of all these species is that they're multiple hosts and multiple woody hosts. So they seem to have the ability to infect and cause root rot and cankers in woody hosts. Every single one of them can do that. So I'm getting towards the end of the talk now, and just a few take-home messages is that we've got really good data for Phytophthora and natural ecosystems in Western Australia, and that is because we've had the Vegetation Health Service here for 40 years doing dieback mapping within the natural ecosystems. We're very lucky to have that data set. It's quite unusual. We've got less data for agriculture because it's tended to be that people assume that, like, if it's Phytophthora, they know what to do with it. They don't need to get an identity on it. Or if they find Phytophthora citrophora on citrus, or if they find a Phytophthora on citrus, they'll assume that it's a species and not actually do a molecular identification on it, because the morphological features are so similar, you can't actually tell them apart without doing the molecular identification. But we have done quite a lot of sampling in the last couple of years, particularly in orchards, to try to increase the data that we've got for agriculture. Most of the clearly introduced species are cosmopolitan with a global distribution, and all the introduced species found in natural ecosystems cause disease in woody plants. Most of the, and I didn't go into it because I didn't want to take too long, but most of the species that we're considering to be endemic are in species clusters. So they're all close. We've got about 4 or 5 species clusters in Western Australia where all the species are very closely related to each other, which is a classic evolutionary expansion in an area, and they seem to be our native species. Urban and disturbed environments should be prioritised for surveys of Phytophthora and all invasive species. And I can do a lot more with the database, and I'm going to be spending a little bit more time on that in my spare time over time. And the next interesting thing that I want to move into is actually looking at in detail at the traits of the Phytophthora species. So these are their morphological traits, their growth parameters, the hosts they've been associated, the types of diseases they cause. So a whole lot of traits that can be assigned to an individual species to see if there are traits that species with limited distributions versus those with broad distribution share. And I mean, there are, but it'd be really good to put some statistics behind it. So we've started with this in a little bit with a paper that just came out recently about the global biogeography and invasion risk of Phytophthora, where this was done mainly on, this was on a global database, so the Australian database plus everything else from the world, which was put together by Peter Scott, where he has looked at every record of every Phytophthora species everywhere in the world, and he has looked at how many hosts they have and how many countries they're found in, and predicted an invasiveness for the species. And now what we want to do is, now that we've got that information, we want to look at the traits of the most invasive species and find out if newly described species have similar traits, because if they do, some of those species may also have the same trajectory to invasiveness as some of the well-known species. So that's where we're at the moment. And with that, I'm going to stop and end the video. I think I'm going to stop sharing. Is that correct? Is that the right thing to do?
Sure, that's excellent. Thank you, Treena. That was so interesting. We've already got a couple of questions coming up. So what I'll do is I'll read them out. In the meantime, if anyone else has anything they'd like to ask, feel free to put that in the question and answer function. Or alternatively, you can raise your hand and we can get to you by allowing you to talk. So firstly, from Simon in Canada. As you mentioned in your presentation, the Swedish study by Redendo et al. Found more Phytophthora in nurseries versus natural settings. In North America, similar results are in agreement with the Swedish study. So my question is, are nurseries the main source for dispersal of Phytophthora species to urban forests, which is a bridgehead to natural forest settings? Would you please elaborate further on this question?
Probably. Yes. So, I mean, this is pretty much the assumption. So Clive Brazier has been talking about this a long time. So the whole concept of plants for planting. So the nursery being the source, so people bringing in plants from elsewhere and through the nursery system, bulking them up and sending them out. I mean, some places have very, very, very good certification of nurseries and some don't. But another issue that we don't really have, that I'm pretty much sure they have, I know it's a really big issue in Europe and I don't know about Canada and North America, is that people can just order living plants on the internet and have them sent to them from anywhere in the world. So then you can bypass the nursery phase if you want to and put them straight into your own garden with whatever diseases they have with them. Yeah.
Yeah, that's definitely a worry, and I've heard of people even getting seeds or plants in the mail without even asking for them. So
yeah, that's happening in Australia. Yeah, that's a big risk.
Next question is from Peter Buchanan. A great seminar. Have you detected in natural systems any native species that have recently become a pathogen of concern on a native plant host?
No, with a caveat. So there is a couple of species that we have described. So Phytophthora arenaria and Phytophthora constrictor are examples of that. Like we're 100% sure that they're native, the way their whole biology is as natives. But what can happen with them is when all the conditions are right, like you've got a flooding event and you will actually see them come in as pathogens in whatever area, like not everywhere, so it's really patchy, so it's not like a front like Phytophthora Cinnamon I have, but you can get like areas where you will get a lot of death, but then you get recovery afterwards. So it's obviously that they're not, so even though they're present there, they don't affect the recruitment of the germination of new seedlings. So probably they're actually part of the ecology of the environment.
Okay, thank you. So from Yupe in Brisbane, she's got two questions. We'll take them one by one. The first is the bridgehead invasion. Is that like the plant kingdom version of the spillover effect in the animal kingdom?
Yeah, I think so. I think it's just been coined as a new term. And actually, the first paper is actually about ladybirds. So it wasn't even about fungi when the first paper, it was Helen Roy and it's harlequin ladybirds in the UK. And I think they followed it either genetically or somehow to show that it had been in the cities that they had built up and expanded before they moved on or something like that. And then it's just sort of been picked up a little bit more by pathologists, I guess, more than anything, or in biological invasion literature, it's appearing quite a bit. And the second bit about it, does it apply to true fungi? I reckon, yes, definitely, because it's always going to be, it's usually the first port of call of nearly everything is either into an urban garden or into agriculture. Yeah.
Okay, thank you. And Kylie Ireland has also said fabulous seminar. Just curious how easily you've been able to interact with collections to update to the new molecular classifications where you have retested those isolates.
Very, very easily. UPay can answer that one even for me. I've been working with UPay. I've been working with Jackie at VPRI. I've been interacting with WAC. And in the DAR collection, they don't actually keep isolates, so I haven't got any additional IDs for them. But it's been going really well. Yeah, we're all working together. And we're writing the paper right now.
That's always good. Okay, and Joanna Wong is asking, are those 5 introduced species that have made their way to native forests in Australia come from the same clade?
No, they don't. So cinnamomi is clade 7, Niederhauseri is clade 7, Pseudocryptogea is clade 8, Nakoshiani is clade 1, and Multivora is clade 2. So they don't. But they all share other traits. So they're all very broad host range, and they can all infect woody hosts. And cinnamomi can produce long-term resting structures within tissue, but all the others produce copious amounts of oospores, which also give them the option of having really long-term resting structures in the soil as well. And Multivora, for example, it's a horrible species. It can survive in calcareous soil. There are very few Phytophthora species that like calcareous soil. So no, they're not from the same clade. Yeah.
Okay, thank you. If there are any final questions, please feel free to go ahead and do that. I have one that's a bit more methodological. What are you finding in terms of the diversity of species between baiting attempts and metagenomics?
I could have gone into that. I was worried I was going to go over it. So I've got a PhD student, Suchana Saka, who is literally doing her completion seminar in 2 weeks' time. And the title of her thesis is, "Why is it so hard to isolate Phytophthora?" And it all came about because we have baiting results, and then we have meta barcoding from exactly the same samples, and you're always getting so much more. I mean, it's one of the reasons why I keep on, like, it worries me, like I'm publishing meta barcoding data, but we can't isolate them. And I keep on wondering, am I publishing absolute rubbish that's just full of, I don't know, like artifacts or laboratory contamination? And so she's worked on this a lot. So first of all, she did a whole lot of work on all the antibiotics that were used in media to see whether they were favouring some species and not others. And she found that there was some preference, but not enough to explain. And then the next part that she did a lot of work on was the rate of sporulation of different species. And what she showed really nicely is that different species sporulate in different times. And when you put 2 species together, so she had to choose ones because if you've got a mixed species, you can't confirm it with a molecular identity. So she had to choose species that looked different when they were growing in culture. She did a whole lot of pairs of species and found, not surprisingly, that the ones that sporulate first take over the baits, and you can never detect the other species, so you just don't get it. So then her third piece of work that she tried to do was in a similar way. She was taking big samples and bulking them, and you'll get lots of species out of the bulk sample, but no, you weren't getting lots of species out of the bulk sample. Wrong thing. Then she tried taking like really small samples, like teaspoon samples, and baiting from teaspoon size samples to see if you would get more, you had more opportunity of isolating less common species by using smaller volumes. And you do, but who is ever going to do that amount of work? So I think that, unfortunately for soil, baiting is the only option you've got to get your Phytophthora species out as a living organism, and it's just really, really hard.
Yeah, thank you. So you mentioned that you were dubious about the initial meta barcoding data that you presented. Are you happy with it now?
Mostly. So I would say, but I'm still concerned perhaps about the few species that we, like the base, I mean, you've got to remember when you're doing meta barcoding, you've only got ITS1 in this case. And so it's not a big gene region, but for Phytophthora, it's actually quite variable. And there are only a couple of species clusters that you can't distinguish based on ITS1. And there's a few species that we detected that have never been, that still, even though there are 100 species recorded in Australia, even some of the ones that we detected in meta barcoding have never been recorded. And so they concern me. And their numbers were very low. So I'm worried that they are artifacts. So what I would probably do now is be much more strict about the number of reads that you have to have of something to include it. So our first lot of work was all done with 454. And with 454 parasequencing, you didn't get as much data. So sometimes you might only have half a dozen reads for something, but they'd be really clean and you'd go, oh, I think this must be it. With Illumina, you get, you know, that countless thousands of reads, and we're just putting the cutoff limit like reasonably high now. So we're not dealing with anything that we get in really low numbers. Yeah.
There's quite a few more questions, and we will have to finish up in a few minutes. So I'll just ask a couple more as we go. So from Falco, can you please talk about how councils or nurseries can try to manage Phytophthora to limit their spread?
I think, well, I mean, a lot of councils, nurseries are, I mean, there is accreditation for nurseries, and would be up to the government to make accreditation compulsory. I mean, but it's not compulsory. So I really wish it was compulsory, and there's a lot of people that know about, you know, the issues with what's being spread around in forestry. So if it was legislated that accreditation of the nursery is compulsory, we'd have a lot more control about the nursery side. Councils just need to be really careful about where they source. So I would suggest, I mean, a lot of councils run their own nurseries for their revegetation plan. That's probably a really good idea. Then they've just got to make sure that they run a clean, you know, nursery, and you just follow protocols around hygiene. And if you're buying them, I'd probably get them tested and get somebody to test them to see if there's any Phytophthora there.
Excellent. Okay, and we'll do one more question relating to your study or the paper you presented. When baiting, was there any, or previously, have you done water baiting for
Yes.
streams and swamps in native vegetation?
Yeah, we have. There was a big study that Daniel Huberly did ages ago. And yeah, we get loads and loads and loads of species, and a lot of the ones that seem to be native to Western Australia have been recovered through the water baiting. Hi, Ros.
Thanks for your question, Ros. Okay, we've got Whelan Mayer in the chat who's asked, what is your cut off for reads to be taken into account?
It depends on the run. Yeah. So, I mean, it's sort of interesting when I'm doing something like general fungi, or we've just got a bacteria, we make it quite high because you've got thousands of species. It's just like a pain in the ass trying to check the taxonomy of them all. I actually, when I'm doing Phytophthora work, I end up taking the ASVs because there's not thousands, there's probably hundreds. I actually always do a phylogenetic, and so I take them into a phylogeny and I identify them based on a phylogeny. And so if I find something weird, so something that doesn't match to an existing species, I go back and look at how many reads there were for it, and is it a funny, I just go back. I check the things that are unusual really carefully and pretty much leave in everything else. Yeah.
All right, well, thank you so much and for all the great discussion points there. I think it's probably time to finish up though, give people chances to get to whatever meeting they've got on next. So if you all join me virtually in thanking Treena for a wonderful presentation. We look forward to seeing you all next. We've got Jeff Powell lined up for our next webinar in August. in July. So thank you so much for joining us, and we'll see you then.